question. All right. Okay. We are ready to go. So hello, everybody, and happy first day of fall. My name is Jenny, and I'm the coordinator for the Metro Phoenix EcoFlora project. Thank you for joining us. Like I said, please remain muted until it's time for the question and answer session. Feel free to type in the chat at any point, and we'll get to those later. Everyone will receive this recording in their email, and it will also be posted on our YouTube channel. For those of you not familiar with the Metro Phoenix EcoFlora, we're a community science project documenting urban biodiversity using iNaturalist and encouraging the naturalist community through events, resources, and more. Each month we have a new EcoQuest, which is like a hide and seek game for different species. And this month we're focused on 10 species of common monsoon weeds and the abundance of butterflies and moths we've been seeing lately. So what are often viewed as weeds can actually be native plants that support these flying friends, and they can also have medicinal and edible uses that are known thanks to indigenous knowledge systems. Besides the 10 plant species that we've observed, there's been about 120 different species of moths and butterflies that we've seen. In total so far, we've had 1,325 observations. So thank you to everyone who's been out there making observations. And I'll put a link to that in the chat in just a second. So to help you feel more confident in identifying plants like the ones in this month's EcoQuest, tonight we have Lia Clark. Lia is a freelance botanist with formal training in plant anatomy and physiology, field botany and collections, and focused research, including but not limited to floristics, rare plant research, plant propagation and seed collection, preservation and germination. Lia is also an artist, currently in a Master of Fine Arts program at Arizona State University, specializing in wood sculpture, and has over a decade of Southwest vegetable and flower gardening experience, basically everything plants. Lia, we're so excited to have you here, and welcome to EcoQuestions.
Thank you. I'm excited to be here too, to go ahead and get this kicked off with some formal definitions and kind of ground us in what we're looking at, you know, when we go around and see plants on the ground, plants in the wild. Just kind of take a little deeper dive into their parts. So I'm going to go ahead and start here, share my screen. And there we go. Is this just showing the, you guys, is it just showing the, just showing everything?
Nope, you're good. Yeah, just
okay, just the presentation. Excellent.
You're
okay.
On the side, so you're good.
Okay, perfect. So I'm up there. Great. Okay, so what I want to do is get you. I mean, I know that everybody here is like very interested in plants, and we have different levels of like experience with plants, experience with ID, experience with hiking and getting in the field and things like that. But I just kind of want to give everybody a little bit of a template or a basic foundation to, it's like a mini botanist like crash course, and also just give you some tools to maybe when you're out there to have in the back of your brain to practice. So that you might be able to identify something on your own, or with the help of someone else. And so I'm calling this Botany Basics and the Observer's Toolkit. And yeah, so let's go. Yeah, so we'll start off with I'm just going to kind of ask you guys a question, and we'll do like a rapid fire in the chat real quick, and just give me your ideas and answers on that, and then we'll run into the science, the plant anatomy and morphology, and the different parts and what they look like and what they do, and probably not what they do as much, but you can ask me at any point what they do. And then once we dive into these characteristics, these parts, we sort of kind of use them in a system as botanists to be able to make educated guesses on what plant it might be. So we'll have some background of these characteristics. We'll have some familiar like family, features we'll go over, and then we'll use those in a system to try to ID. And then we'll go over some tools that you want to go out in the field with, or you want to come back and use to figure out what plant you have. And then also maybe you want to talk to some people, but we'll get into that. So we'll just kind of wrap up with the tools and resources for identification plants. Okay, so I'd like for you guys to go ahead and just answer this question and just kind of shoot them at me in the chat. What is the first resource you think of when you're trying to ID a plant? Shoot. Okay, iNaturalist. All right.
To be fair, the audience might be biased.
A little bit.
Laura.
Little bit. Yeah, no right answers here. Just, yeah. My eyeballs, great. Google Lens, iNaturalist. Okay. Nice. And Epples Plants of Arizona. Great. Oh, we've got some informed people. Google. Yeah, sounds great. So I'm definitely going to go over some of those today, and I really like the Google Lens thing because that's been really accurate lately. And friends, yes, great. So that was more for my information, not yours. So thank you very much for that. And let's see, can we go back? Okay. So September's EcoQuest is, we're looking at weeds around here, trying to figure out what they are. And when you're asking about a plant, whether it's in nature, in the city, in your house, in the nursery, you kind of want to figure out, you want to consider where you are. Does it belong here? Have I seen it before? Or do you think it belongs here, right? Because we don't know that answer quite yet until we identify it. But is it something that you've heard of that's invasive? And then what are its features like? Is it a shrub? Is it a tree? Is it a really showy flowery type situation? Is it a small herbaceous annual or perennial? Got the little, can you guys see my cursor at all?
Yep.
You can see my cursor?
Mm-hmm.
Great. So, this is out in Botany Mountains, Botany Peak, and, in the city, got some weeds just hanging out. I know you've seen that one on the right there before. And then, different habits of plants, bushes, aquatic plants, where am I? What am I looking at? Does it seem like it belongs here? And let's look at those parts that they have. Because to be able to identify what you have to look a little closer. So let's dive right into, as if we were going into a botany course. You're speed dating me right now, like we're speed dating. And so if we're picking apart a plant, there's a ton of separate pieces that do different parts that do different things. And down here, we have your root system and your shoot system. We have leaves. We have a flower, and then you have just the whole habit. So we kind of separate it into an above ground and a below ground system. And there are a little bit more technical terms here. You have the petiole, which is the stem that goes from the stem to the main stem to the leaf. Then you have another like, and then in here, you might have buds. Maybe they'll make another shoot later on. And then nodes and internodes, just places where tissue develops. And then you have sort of like your apical, kind of forever growing, unless you chop it up, which it might come up from the lateral buds, but kind of indeterminate growth growing straight up and out. And then you have your flowers, okay? And I think a part of identifying that, one of the bigger parts of identifying plants has a lot to do with their flowers and their arrangement and their color and their shape. And so you don't have to know this, memorize all this right away, but let me take this down. You know, we're going to have to start paying attention to, if we want to ID, we're going to have to start paying attention to how many number of anthers are there. You know, what is the style shaped like? So we've got a few parts to that stigma style over. That's the female part, and that's called the gynoecium. And then we have the male parts, which are the stamen, and those are the androecium. And that's where the pollen goes. We got the pollen fertilizes it lands on the stigma, travels down, fertilizes the ovules down here, and then we make a fruit, right? They didn't put it on here on this, but the stem that goes to the flower is called a peduncle, or yeah, a peduncle. So let's see. So I'll start going through each of these here, just a little bit more in detail. I wanted to run through roots and shoots real quick because we don't do a ton of identification based on roots and shoots, but you know, every little bit helps. And so the main types are fibrous roots, which you have just like a big kind of net situation happening here on a grass, or happens a lot on monocots, happens a lot on a lot of plants, but monocots tend to be a little bit more fibrous. And then you have like a California poppy type thing here, and it has a big fat taproot with some kind of fiber-like roots coming off. So, and then there are a couple of other things like aerial roots. This is a picture in my greenhouse of a monstera stem that is just growing aerial roots like crazy because it's at about 80% humidity all the time. And then this is a picture of a fig that has buttress roots, and those are for stability. All the roots are for stability for the most part. Even aerial roots will cling on to trees or anything like that and provide stability. But here are all the different types that you can imagine. So leaves and flowers and seeds somewhat are going to be your main features that will sort of diagnose what plant you have. We now have DNA sequencing, but for a long time we didn't, and we spent a lot of time looking at these little different features, and there's that whole system of there's a whole gestalt to it. And so when you're looking at leaves, you want to pay attention to you know, what size they are, what's their arrangement on the plant. So you know, on this one, you know, they're opposite, okay, of each other. You can't really tell as well here, but they're opposite. You know, they come off of the stem at the same point. And say on our amaranth, you know, then that plant, the leaves start coming off of the stem, one after the other. So they don't come off in pairs, they come off individually. So that's called an opposite and alternate arrangement. And then you want to pay attention to their shape, okay? We have like an oval shape, ovate shape. This thing is in the way. We have like a lanceolate type shape that is not on here, but it's basically like straight. It's like this one, but a little bit longer. And then we have like palmate, type leaves that are like, they come in fives, the shape of fives. We have leaves that are pinnate. So there's all these special terms for all different types of shapes. And this is actually from Harris and Harris Plant Identification Terminology, which is a wonderful book, wonderful thing to have in your pocket right away when you're trying to ID a plant, because if you're keying or if you're looking at some kind of descriptor, then they're going to throw a lot of these official terms at you and you want to know what they mean. So I'll recommend the book at the end of the talk if you don't know it already. This is a picture of Venselia farinosa, native here, and this one has it's gray because it has a ton of trichomes on it, protecting it from the sun, has a boundary layer to keep temperature regulation, and so we call that pubescent. And then like a smoother type leaf here on this philodendron is glabrous. So you can see it's real shiny and smooth, and this one belongs more in the jungle. A lot of water comes off of it and drips down. So you don't want to, if there's water all the time, you don't want to retain it for too long. The margins here are really fun. So you can see that the philodendron is quite lobed here. The monstera in the back has an entire margin, but it's also, a fun feature of fenestrations on the leaf surface there. And you think about trees that are native here, prosopis and mesquite and palo verdes and stuff. They have, these pinnate leaves, but they could be pinnate and also like have subsections of leaves that are, pinnatifid, and so lots of variation. And sometimes leaves can look really similar or really different. I don't have a visual example here, but if you know what globe mallow is for Elsia and the weed mallow that we have out here, we've got way different leaves, but they're in the same family. So you can have large variation on the leaves. They can be really, really similar. And that's why we definitely want to consider more characters of the plant. So we move on into the inflorescences. Oh, we're missing a picture there. I don't know why it didn't come up. So we'll move on to the flowers and the inflorescences, and you'll often hear, like, I don't know if anybody goes into different ID groups seeking advice and then show out like a cactus or something, and it's in this genus where all of them look the same but their flowers are different, and you show them a picture and the most of the part of the plant is there but they don't have any flowers. If it's an expert, they're going to say, I need to see the flowers. So they're really, really indicative of the species. Flowers probably the most. Seeds can also help with that, but seeds can also help with figuring out what family it is, because a lot of times, a lot of the seeds look similar in the family. Maybe the flowers look different, or something like that. So we kind of want to figure out this like gestalt. It's an art form. It's what we've had for hundreds of years before we had PCR DNA sequencing. So I just wanted to show you some different types of inflorescences. You got some simple inflorescences. That's where we're talking about the pedicel that leads to the flower. And you can have, flowers. These dots represent the individual flowers, and you can have them come straight off the pedicel, or you can have them come branch off of the pedicel individually, or you can have them branch off in clusters. And that's your typical spike, raceme, and panicle. We've got a penstemon here. Okay. We've got a penstemon here showing basically, let's see, a little panicle. So because it's got a stem coming off in multiples. And then we do have a cyme, which is similar, just kind of like and there can be different kinds of cymes. We do have some lovely plants that have, like here in the desert, annuals that have like different shaped cymes. One of them is a scorpioid cyme, and that happens with heliotropa and bluebells. And then there's this head situation. Composites like sunflowers and anything in the Asteraceae, they usually have sort of a head type situation. There might be a few other things, but they have this very large gathering of flowers on a whole head, basically. So you got this big thing that looks like a flower, but it's kind of like many different kinds, a couple different kinds of flowers and many thousands of little flowers in there, So it holds many individual flowers. And then you got this weird guy, Q. They reference it as a spadix here, but the spadix is the outside here and the spathe is the inside here. And that's that big stinky corpse flower that blooms once every, 10 years or something like that. And that's pretty cool. It's a monocot. And it attracts flies and dead body smelling thing. That stinks pretty bad. It attracts flies and things that eat carrion and stuff like that. Grasses have flowers too. They are very, very tiny, and it takes a very, very special person to study grasses. And we have a very special person in this state that is nearly a grass expert. And but I mean, they don't look typical at all. You don't really see very many petals, dealing with a lot of bracts and awns and just weird things. Grasses are so weird. And there's a picture of, yeah.
Are you talking about Liz Makings?
Yeah.
I just want to throw in that next month we're going to have her for EcoQuestions. So if
excellent.
everyone wants to learn about grasses, tune in next
month.
month.
Man, she's going to show me up, man. Okay, yeah. So she's going to totally let you know all about the weirdness of grasses, because it's just like, you think composites are a big family, and over here, which is Asteraceae, are a big family, and they're a pain in the butt, but you don't know nothing. Grasses are, yeah. So there's a picture of wild oats, and each of these individuals are fruiting by now, but they have their flowers in there, and they're wind pollinated, and they do their thing. And then you have something like, I don't know if you guys have seen this in your garden, or if you do any vegetable gardening, but something like dill or carrots in the Apiaceae family. They'll make these stalks, these flower stalks, inflorescences that are like umbrellas, okay? And this one is actually an umbrella of umbrellas, okay? We got like 7 little umbrellas here on a big umbrella. It was kind of fun. Queen Anne's lace, I don't know if you've ever heard of that. But these are individual umbels on a compound umbel. So you'll run into a lot of these different kind. There's a ton in that plant identification terminology book. It's a lifesaver. It has everything. And sometimes even a little bit overwhelming. So just, you know, these are kind of your basic morphologies for inflorescences to look for. You know, start going out, and if you're seeing something flowering, you know, take your book with you and see if you can identify what kind of inflorescence it is. What's the shape of it? Look a little closer. How many little stems do you have there, or peduncles do you have there? You know, kind of get yourself more familiar with the arrangement of the flowers. So we'll get into a little bit more into the flowers. I'm sorry I don't have any flowers on the side here to show you, but I wanted to concentrate a little bit more on how we use these to determine, use flowers to determine we use them for like family characteristics and to identify maybe what family. They often have like trends, family trends, where, you know, there'll be 6 anthers and 6 stamen and, you know, 1 pistil, or 5 stamen and, you know, 1 multi-lobed stigma on the words. Look here, style, the andrecium and gynoecium. So this is the gynoecium, female part, male parts, andrecium, okay? And those are the official like science words. Some people are like, they don't like to call it male or female. They don't like to gender it. It's, even though in Latin it's still a bit gendered. But yeah, the whole pistil here is the merging of the stigma, style, and ovary. And the ovary contains the ovules where the seeds will develop. And yeah, so you want to know, okay, how many petals does it have? You know, this one, if it's bisected, probably, you know, 1, 2, 3, it probably has 6. You know, bisected, cut in half, it has 6. You know, it's got a weird number of, I don't know, 4, 8. You know, this is just a generic, right? So we've got like 8 stamen here, and basic, simple style, simple, you know, nothing, you know, crazy pistil. And you just want to note these things when you're out in the field. Write it down, take pictures, and so you can go back to your references and compare. Okay, so you can do a little bit more IDing with seeds. Like I was saying earlier about, you know, sometimes species in the same, or genera even in the same family, will have very similar seeds. And you can use that information to help determine what it is. But, you know, they can be really specific, but they can also not. So we tend to focus on the flower for more of a positive ID, because there's just so many more characters you can examine to liken them to the family. Also, I underline quick note on seeds because I love seeds so much. Seeds are my life, and I will go on and on and on about seeds. So I'm gonna cap it there because they're awesome. So yeah, you know. So now that we're familiar with what we should probably pay attention to in some of the characters of a plant, there's rhyme and reason to all this. And botanists use a system developed by Linnaeus way back when, where we kind of, you'll see, you know, when we talk about in scientific names, we'll talk about two names. So for example, down here, Portulaca oleracea, okay? This is a taxonomic, this is called the taxonomic rank, and all life is divided into a scheme of this sort of scheme. And we have some examples here on the side, and it goes from most general to most specific. And like it says here, increasing in similarity, domain, phylum, class, order, family, genus, species. Okay, if you're like a avid amateur or intermediate, you're probably going to go up to family. You're probably going to know a genus, maybe some species. We're trying to figure out like probably up to family characteristics here. If you're a little bit more, you study taxonomy or you study the history of how plants evolved, you're probably going to deal in family, in order, kind of hang out in here. And then it starts getting, a little bit more general, in here where we start talking about not plants, we start talking about fungi, and we start talking about animals and stuff like that. So on the left here, you can see, this is the Eukarya chordata. This is us, right? Homo sapiens down here. And then we belong to the genus Homo. There's also, what's that old fossil?
Lucy?
Yeah, Australopithecus? Is that what she was?
Sure. I
think she was Homo, well, there's Homo sapiens, Homo erectus, Australopithecus. So, and then, right here where we see primates, we might relate to others, primates like orangutans or gorillas or something a little bit more general. Same, well, we got bacteria over here. These are your good gut bacteria. I can't tell you anything about these. I'm not a microbiologist. I pretend to be sometimes, but I'm a plant biologist. So we're mainly going to be dealing with genus and species when we're talking about nomenclature and naming plants. A little bit of family. Or sometimes I notice we'll go out in the field and we'll just be like, maybe we won't be in our home place where we know everything as botanists, right? I'm a creature of the Southwest and know a lot about desert plants, but send me to Wyoming and I might be like, that belongs to this family. I don't know, because of these characteristics that are similar, you can tell that, oh, it belongs to the Malvaceae family, or it belongs to the Asteraceae family, but you make an educated guess, but you don't know exactly what genus or species that is. So, that's again part of the gestalt of getting your botanist wings. These are the kinds of things you want to start guessing and understanding with practice. I put this little PCR DNA sequencing thing down here because it's kind of like, there's this, we've been using this binomial nomenclature system for forever. And that's all we've had until, the 20th, 21st century, where we start learning how to sequence DNA. Rosalind Franklin, with the work of Rosalind Franklin and Watson and Crick, they've come up with figuring out basically the language of all living things. Their PCR DNA sequencing has allowed us to figure out exactly what's related to what. It's often more accurate than what we've relied on for a really long time, what I'm trying to teach you here. And ultimately, you know, us morphologists have to bow to DNA sequencing because it's a lot more accurate. But for now, you know, not everybody has access to DNA sequencing all the time, and it's expensive, and you know, you got to know how to use it, and you got to know how to read it and stuff like that. So what we're learning here is really nice for anyone who wants to get into it. And it's been a really good system for a long time. So I imagine some things will change in the future. But for now, we're still doing a lot of approaching identification through these binomial nomenclature and family characteristics. Okay, so we'll get into a little bit more on family characteristics. I'm just gonna run through a family on one of these weed plants here and see, and let me know in the chat later if it's clear. I just have one example so far, but we can run through other examples if you want in the chat, because it's a lot of information. And I mean, you guys are doing years of what I've learned in years in like 30 minutes. So that budding botanist thing was nuts. I'm trying to simplify it even more. Okay, so if we're gonna start thinking about narrowing down in a system, there's like major groups of plants, then there's like families of plants, and then, you know, genus and species. So we're kind of functioning in the taxonomic rank. And one thing that it comes to when it comes to IDing plants on a general level is there are monocots and dicots. And monocots will be like your things like palms and grasses and jungly vine type things. And what they do is, in their development, they only push out one leaf at a time, okay? In their embryonic stage, they only have one leaf that develops. And in dicots, there are two. It's always paired, whether it's opposite like this or not. You're always going to get two cotyledons, and a cotyledon is basically it's the first two leaves. Okay, let's think about back in, you know, 4th grade when we sprout some bean seeds. You know, you get these first two leaves that don't even look like, you know, they just look like big chunky fatties, and what they are is a food storage system. And so that's their primary function. They're not here to specialize in photosynthesis. They're here to give nutrition. So they're going to be big, chunky, fatty, and they're going to shrivel up when they're done. And so that then leads to the first true leaf, and the true leaves will start looking like, you know, what the plant is indicative of. But yeah, you want to figure out, okay, is the growth point, or is the habit expressing, you know, growing one leaf at a time or two leaves at a time? And then here's some characteristics that we can look at all over the plant between monocots and dicots. This is a highly specific, so we're not going to do, I'm not going to quiz you guys on pollen grain, difference in pollen grains, because, you know, we only have dissecting scopes and loops. We don't have compound microscopes. So like I said, in the seeds, well, they'll have one cotyledon in monocots. So here's a little embryo, okay? Here is the, it's like the leaf progenitor. Okay, so it'd be just one. And then in dicots, we have 2 cotyledons. We also have 2 leaves. And then in the flowers, this is really important. You want to pay attention to, like, where are we in the number of petals, all right? Where are we in the number of stamens, just like I was mentioning earlier. And so dicot, oh, I've got this backwards. Okay, I have got this backwards. Why didn't you tell me? Okay, so just ignore this right here. So we have dicot here and monocot here. And dicots usually have maybe 4 or 5, in multiples of like 4 or 5. I mean, they can, like, petals or sepals. So petals are, you know, we've got petals, and then we got sepals in the back, which are kind of like bracty petals. Those will be in multiples of 4 or 5. The stamen will be in multiples of 4 or 5. And in monocots, they'll be in multiples of 3, 6. So you'll want to look for, you know, count the number of petals and sepals that you have, stamen that you have. This is the characteristics of the leaves, and while this is generally true that the leaves are oval or palmate, and the veins are net-like for sure, and then the leaves on monocots are narrow with parallel veins. It's not always true. You'll notice in that, okay, I'm gonna change slides really quick. You'll notice in my greenhouse here, this is a monocot, and it grows 1 leaf at a time, and it's certainly not you know, super elongated. This one down here is a monocot, so there's a lot of variability in leaf structures, but, you know, take it with a grain of salt here. Not going to quiz you on vascular bundles, because this is a morphology situation and not an anatomy situation. So morphology is like the whole plant, the sort of macro size of the plant, all the things we can see with our eyes, and anatomy is more the inside parts that have to do with the cells and the vascular arrangements and the layers of cells and the leaves or things like that. So I'm not going to quiz you on that, and not going to quiz you on taproots or fibrous roots too much. These here are just kind of like, if you're looking in the book to try to figure it out, you just want to kind of make the checkbox that it has fibrous roots, or make the checkbox that it has taproots or something like that. Hey, Jenny, I put it on for 29 hours, not 30 hours, not 30 minutes. So I don't know if you're keeping time, but
yeah, we're at 7:20, so like 10-15 minutes ish.
Oh, okay, let me get moving. Okay, so then we'll run into some, I was just talking about, like, trying to identify different characters, and there's going to be like a lot of different sizes of these characters. Some leaves might be really tiny, some might be really big, some flowers might be really tiny, or like you can't even see them at all. Some can be really big. So when you go out in the field, you're going to need some tools, some basic tools, and some light stuff that you can put in your backpack. And then when you gather your data, you're going to need some tools to evaluate them. And here's a picture of me looking at some equisetum. I got my digital camera there to take a picture of what the plant looks, but also geotag it from where I'm at. We've got, you know, my pencil and notebook. It's really important for documenting like what's around you. Oftentimes you'll get clues about what your plant is by what it's associated with, by what plants are around it. If it's, you know, you're on a hike or something like that. No bars in the city, because Phoenix is a hodgepodge of all kinds of ornamentals, and you know, just wouldn't be able to narrow it down. But if you're in nature, in like a preserve or going on a hike, there's a lot of associated species that might help you figure out what you've got, because they occur in the same area. It's all an ecology, it's all a system, and nature's got it all figured out. So write down your, you know, your details. You can also write down what kind of animals are associated with it. If you need to see some tiny parts in the field, grab yourself a jeweler's loop. This one's a 30 times, but you know, a 10 time is standard. You know, and then you have ones that are like, you know, all three. You know, you can have multiple magnification on one jeweler's loop. This guy up here on the left is sort of misleading, but it's a dissecting scope, but it's you can plug it into your phone or you can plug it into your computer, okay? And the misleading part is here that it's telling you it can see cells. Not exactly true. It can just do really, really good close-up of small features, like on the flower, you can see the stamen and the stigma better, or you can see some little bugs better. You cannot see on the cellular level that well. But it's really neat. These are really affordable. They give an okay quality, picture quality, but if you need to see something on the fly, bop this into your computer in the USB and bam, you know, you don't have to spend. We've got a Liz over here on the right-hand side. She's got like a real dissecting scope with, you know, a light ring, and this is what we use this is good equipment. You don't have to go, I mean, you can get them now for, I don't know, I just got one, it's like $400 or $500 for a good dissecting scope. But I mean, this guy over here, 30, 40, 50 bucks, plug it into your computer. You know, in the field, you know, you might be able to magnify pretty good. They're really neat, really neat. I found out about those first in 2014, and it made having a dissecting scope accessible to me as a person that historically doesn't have a lot of access, resources, or money. And so I just got really stoked about that. But dissecting scope, you know, you hear the scopes in school, you know, you look at a compound microscope where you can see all the cells, and biology class, you prepare a plate. We're not looking on that level. That would be an anatomical level, anatomy level. We're sticking in the morphology level, and a dissecting scope is good enough for that. Really you can see really clear, really beautiful images on it. And this is Liz at the Arizona State, those are my glasses, Arizona State University Herbarium, looking at a collection I made of Eryngium sparganophyllum, and she's tearing apart each of these inflorescences to get some fruits out. And fruits not synonymous, fruits hold seeds. And here's a little Eryngium sparganophyllum seed, which I just think looks super cool. It's like a little pangolin or something. And on the other side, it's scaly, you know. So it looks like this on this side, and it's like scales on the other side. Really neat. And, you know, these things are tiny. You would smaller than a grain of rice, you know, less than half the size of a grain of rice. You can't tell all this beautiful texture, you know, without a scope. So, you know, you might not be interested in getting that deep into it, or maybe you are and don't have the money for that. You can always hang out with us botanists, you know, or schmooze us and see if you can use our dissecting scope. So they're available if you want to volunteer or something in your bariums or anything like that. So they're really fun devices, really cool technology that I love. I love microscopy on all levels. Okay, so those are some of your tools. Hope I didn't miss anything. Okay, so now that you've got tools to evaluate those things, I was talking earlier about separating the characteristics of the plant, or distinguishing what the characteristics of the plant are and seeing if they fit into like a family scheme. And this is
Hey, Leah,
yeah,
just want to give you a 5-minute warning.
great. This is Wendy's. This is great book, wonderful book. This is sort of mid-tier, and I'll show you some resources of what you can do as just kind of like a beginner. But this is sort of mid-tier botany and identification. And she's done all the work for you in identifying the characters of a family. And so thank you, lovely lady from Georgia. And so this looks daunting. It's a lot of information, but I'm going to break it up here for you. And I chose the Amaranthaceae family based on some of the weeds that you guys are looking at this month. And, you know, here it just gives a introduction of the different characters and what do they have in common? You know, what are their leaves like, or what are their flowers like? What, you know, they show here 2 different plants in the Amaranthaceae family. So they can differ in habit. They can differ a little bit in the way that they flower, but they have generally these similar attributes. Okay, and this is just kind of all the information telling you about that. And, you know, with all the technical terminology that you can look up in your identification book. So I'm just going to run through one of them. And this is the image from the blog. And this is an Amaranthaceae family. Okay, pigweed. We see this popping up all over the city after the rains, popping up every summer. It's also a superfood, and you can harvest it and eat it. But you know, when it talks about the habit, which is the general shape, says herbaceous to shrubby plants with reddish stems. Okay, can't see that much here, but herbaceous, you know, a little bit shrubby, could be smaller than this one. You can't see it too well here, but they do sort of turn red here, and you'll see different varieties. Like this one is really red on this side, but they tend to give a little bit of a red color. And then the flowers, okay, so they have dense cymose inflorescences. That's what you're looking at here. This is a dense bud of flowers, and it's got tons of tiny little flowers, make tons of tiny little seeds, and subtended by scarious bracts. And here is sort of a blow up of one of those flowers. And, you know, it'll be an androecium of 5 stamens. So what do we remember that was a dicot? You know, it's either 5 or 4 or 5, or monocots were like multiples of 3. The gynoecium is of 1 pistil. It could be 2 carpellate or 3 carpellate, meaning the sections, we won't quiz you on that. But, you know, there's usually just 1 pistil and 4 or 5 stamen. Okay, so I'm hoping that is getting through, and you can ask me more questions about that in the chat. But, you know, most of the plants in this family are going to have these characteristics. And here's a good example. Okay, so let's get back to the beginning when I asked you about like where you want to go. A lot of you had to figure out what plants, what your plants are, and a lot of you had a lot of good, you're, you know, already up there figuring out what to do. Google has made things amazing for just trying to figure out things right away. You know, when we ask, it's like, oh, I'll Google it. And what will you say in the Google? Small, herbaceous, or small, showy, red flowers, bush, or something like that. And that's a perfectly good way to, you know, try to figure out what on the fly, what your plant is, but maybe not necessarily very specifically down to the species. You know, so you can say, hey, it looks a lot like this, or, you know, it looks exactly like this. And then one thing I noticed was Google Lens is getting really, I've spent a lot of time in these like plant apps, and like it was not going well for a really long time, and Google just knocked it out of the park. It's not accurate all the time, but it does a really good job of, you take your picture, you throw it in Lens, and it uses algorithms and all the data that Google has, which is tons, and tries to match it up. And, you know, but we want to kind of get you out of Google. I mean, it's really easy. I'm not, and it's a great resource. I'm not like asking you not to do it, but I would like to illuminate some other resources. If, say, you don't have your phone, say you don't have Google, so if you're out on a hike in a particular area that has a visitor center, I have Oregon Pipe National Monument down here. A lot of times when you go in the visitor center, you'll they might have a species list, or they might have a botanist there. Not so much on the latter, but a lot of times they'll have a species list of what's out there in the area, and you can just ask them, you know, and you can ask whoever's there also what's in the area, and write it down and look it up later. And they're really good resource. When I go to a new place and it has a visitor center, I'm always grabbing the list and going out there. One of you mentioned field guides are really great and really available to just anyone as far as the literature goes. There are books that are just like a lot more dense and difficult to get through, but like this like Plants of Arizona field guide, it's really easy. It arranges things by flower color. So you can just pop open the book and see what flower color, and then just like, you know, start narrowing it down from there. Really beautiful pictures all the time. That's part of the draw to it, is it'll arrange it by flower and show them really well and beautiful. And it's something you can carry in your pocket also, or in your bag. It's a little bit bigger than a pocket. So this stuff was kind of like, I'm by myself, I'm in the field. What are my resources? This is sort of more of a crowdsourcing type slide that you can take advantage of. When you gather your information, you can also go to experts at the Cooperative Extension. University of Arizona has one here in Phoenix, and they're really good at identifying, you know, the plants, but they're also really good at identifying insects and pathogens. They're just kind of like jack of all trades for identification. And you can take a specimen down there if you want, if you have a specimen, or if you have a picture. If you're in more of the city, you're gonna be dealing with a lot of ornamentals. So go to your nursery. Go to a mom and pop nursery where they know what plants are which, and say, you know, I have this, my neighbor has this one, I really like it, what's its name, I want to get it, you know. You can talk to your friends and learn from there. Even, you know, now with where I'm at, there's a group of people that I still, that I also consider my friends, that I still tap their knowledge as well. You might be familiar with Kara Barron. She did also one of these EcoQuestions sessions. And this is, we were doing a finding milkweed at Mount Lemon, and I just learned a whole bunch about the specific type of plant when I was on this outing. One thing that I really enjoy, once you get past all the drama, right, because it's Facebook, but I really learn a lot about something I don't know much about right away in Facebook groups. I mean, not everybody's an expert, but they're dang better than knowing nothing. And it's a great social way to talk about, get your, what is this? What plant do I have here? And why is it doing this? And sometimes in these groups, you will have experts, and they will chime in and do an ID for you, which is fun. It's kind of nerdy thing for experts to do. We like doing it, and then we don't, and we do, and we don't. And we'll chime in on our meantime and just be like, that's of this and that's of this. And it's good practice. So don't ever feel like weird about asking, because we like to do that. It's nerdy.
I'll vouch for that too.
Yeah, and it keeps the mind fresh. So it's like, yeah. So there's several groups you could join. I mean, there's tons of groups, but here's a few. The Native Plant Society, there are different chapters. Kara's got this, is part of this Gila chapter. There's a group called Arizona Native Plants and Plants Identification. I mean, you could write to that one for our purposes. If you want to identify houseplants, Arizona Houseplants Enthusiasts, Sonoran Desert Wildlife, there's tons. So, and then there's also iNaturalist, which is freaking amazing as well. It's like, so we have, as professional botanists, our own databases that we really have entered a lot of verified scientific information into about plants. But it's really valuable to have the plant world more accessible to more people. And iNaturalist did a really good job of creating an app where people could just go out and document things. And you can do it right on your phone. And you can also get your observation checked by a professional, a research grade professional. And it's a fantastic resource. It sounds like a lot of you are aware of it. And then, okay, so now you can get into the nitty gritty, right? You could really start like keying stuff, right? Figuring out how to key, which is like sort of kind of, besides PCR, DNA sequencing, keys are like the ultimate guide to really narrowing down to exactly what you want, or exactly what you're trying to find. And we're not going to go into keying, but it's like the Bible to figure out exactly what plants you have. So that's Arizona Flora. And the web resource that I was talking about, where some of you know, where we store all of our observations and collections, and is SciNet. And I mean, there's other different, this is for, Arizona, New Mexico's for the Southwest, and some of the little bit east from here, but there are other ones for like, the tropics and Europe and things like that. So really great resource, a little bit clunky, and a little bit harder to figure out than, say, iNaturalist or one of the field guides. That's like mid-level, but totally accessible and free and awesome.
Hey, Leah.
And yeah.
Just want to let everybody know that we do sign it workshops. So if
Yeah.
you sign up for our newsletter, you can stay in tune with those. And also, I'm going to ask you the one question that's in the chat right now.
Yeah.
So just in case that person wants to
I
head
think
out.
we have, yeah, we have like one more slide.
Okay, cool. How do people volunteer at an herbarium?
So perfect. I'm glad you answered this question because that's this slide right here. So so you've got, you know, resources, right? People and organizational resources. And one of the places that does it is that deals with plants and identification and collection is a herbarium. And locally, we have Arizona State's vascular plant herbarium, which I worked in for a while. And this is the curator. She's not the curator, but she's a manager of the Arizona State vascular plant herbarium. You'll want to go on her. You can. This is clickable, and you can go on her staff page. But I would just email her and say, I would email Liz and say, hey, I want to do a little bit of volunteering. Okay, you got this resource over here from the DBG. We've got our host. Okay, and we've got all these fantastic scientists that I love and are very knowledgeable on a multitude of subjects. Okay, these guys right here, Andrew Salawan and Wendy Hodgson, they run the show at the herbarium at the Desert Botanical Garden. So how you can volunteer is you can ask Jenny to hook you up at the Botanical Garden with like the volunteering there. You know, so you just ask, you look up people and ask at the herbarium and see what they can do. They'll put you in touch with the right people. Hope that answers your question. Yeah, so you've got these resources, and I put together a little like a little library. This is like our as botanist toolkit. These are like all the books we use, and we can make that list available to you in the presentation or outside of the presentation. We can do that later too. But I sort of arranged them in, you know, manner of beginner, intermediate, and advanced, and because sometimes people give out these lists of books and you just don't know where to start. So I just kind of wanted to, you know, hey, start here. You've got the field guide, the flowering plant families, you know, botany for gardeners, botany in a day. Those are things you can start with right away. Those will help you out. But and then the other stuff, if you want to dig deeper, dig deeper. But that's it. Thank you for listening to all of those words. And I'm ready if you have any questions, if anything wasn't clear, if you want to know more. I love answering questions like Jenny said, total nerding out. Let's nerd out.
Thank you so much, Leah. I just want to let everybody know that I've placed a bunch of links in the chat. Leah's contact info, my contact info, and links for EcoFlora. So those will also be sent out with the recording, so don't panic and feel like you're going to lose them when we close the meeting. The chat's gone, but we'll send those out too. I highly recommend, if you don't do anything else, sign up for the EcoFlora newsletter so you can stay in touch about events like this. And yeah, we'll take questions now. Does anybody, that's the only one I saw in the chat. Does anybody want to unmute and ask verbally?
Do it. Do it.
We have a
I'm
lot of thank
excited.
yous in the chat.
You're welcome.
Rodrigo says, thank you. The more I attend events, the more I get info in my head.
Yeah. And that's what we love to do. We just love to share information, information files, full files.
Totally. I'll give it another minute or so. I know that that's
Okay.
a lot. It's a lot to put in your brain. But
Hard to come up with just one question with all the information. Yeah, totally. I got, you know, I need a drink of water. It's a lot of information. So yeah, feel free to, you know, I have my contact information. All the experts are available to you too.
And we'll
Oh, that's what I wanted to say. Okay, so Rodrigo, right? Sometimes we go out in the field as botanists, and sometimes we need help, and sometimes we just want company. And they're like field trips, and like if you just check with your local botanist. Hey, do you need help? Do you want to go out in the field? That you'll learn a lot that way. It's almost like apprenticeship style. It's fun.
I wholeheartedly agree. And we'll also be doing more kind of in-field stuff coming up now that we can do limited in-person. There's the timer. We can do limited in-person events. So keep your eye, like I said, on our events. We'll be going out looking at grasses next month. So.
All right.
All right. Well, thank you so much, Leah. It's been
very
really
welcome.
fun learning from you. You're a great person to learn botany from. And like I said, everyone will send these links out, and Leah's slides will also be emailed with the recording. So you'll have those and you can click all those links in the presentation. And that's it. Everybody, I hope you enjoy the rest of your Wednesday evening.
All right. Have a good night.
Thanks again, Leah.